Welcome everyone to this, I'm calling it the Multiscale Neuroscience Journal Club. I couldn't think of anything better right before sending out the email, but I do think that it ties into what kind of evolved as the kind of interest that we had going from the dynamical systems journal discussion group to the Grossberg thing. And especially like Mac is very good at this, and I'm trying to get good at it, which is like kind of jump from like very detailed stuff about neurotransmitters and receptors all the way up to the system level, and then from there to behavior. And
it just involves this, Yohan. It's just a lot of hand waving.
We're very good at that. So I think actually this paper, I did that little poll, and the evolution one was actually, I think, the winner. So that's why I thought of this one. And I think it really fits because there's almost so much detail that you can like completely fail to like zoom out. But Paul really wants to get people to think about the overall, like basically reframing of behavior. So hopefully I'll have some narrative in here, but it's mostly diagrams. So this paper from 2021, for some reason Google's column has the year wrong, called it 22. He's kind of saying that we have this phylogeny of concepts, or not exactly phylogeny, but a way of thinking about cognition, perception, memory, blah blah, that sort of arose from philosophy. Like it precedes biology in many ways. And so he wants us to go from the figure on the left to the figure on the right, which is sort of more in keeping with what we know about sort of integrated nature of function and the phylogeny, the evolution of different parts of the brain. There's a major challenge involved in the seemingly innocuous sort of goal, which I'll get to. So this excellent figure, which if you want to see him walk through it, the video I sent out has him kind of step through it. But basically, in the paper, he's very careful to say, I'm not talking about the great chain of being. It just happens to be that man's on the top, could just as easily be on the bottom. So yeah, it's kind of like greatest hits in the development of the primate nervous system, or steps along the way to get to where we are. So you have the development of neurons, which were kind of basically loose neural nets. There wasn't really a brain. In animals like, I believe, Hydra has neurons. And interestingly, the centralized clump of neurons, it precedes the heart. So the brain is older than the heart. Somehow for me, it was sort of weird when I learned that. But yeah, so you have this sort of the formation of neurons, and they kind of collect in a kind of tube. And then basically one end of the tube becomes the hypothalamus, and then all kinds of things start to happen to the ancestor of the hypothalamus, and that's the story of these papers. Basically, he talks a little bit about what it connects to. The main focus is really how the hypothalamus sort of explodes, and he keeps trying to kind of bring that back to behavior. And as we'll see, a fairly simple story about behavior. So the simple part is the function side, at least from the storytelling perspective. I would actually argue that it's almost like the outline of one of those big center grants, like to have this plus a lot of people doing modeling, like combined with artificial life. Like you start by making really, really simple models of creatures way over on this end, and kind of work your way up, and eventually give up well before you reach mice. But that seems like the way to really flesh this out in a way that modelers can really kind of almost enjoy the process. So before we get to how he kind of populates this story, I kept thinking about this while reading this paper, that there are some serious problems with attempting this, right? So basically, soft tissue does not leave fossils, usually. I saw a documentary about geology where there were these ediacarans who did leave fossils even though they were soft, so it's not completely true. But yeah, so it's really hard to know what the ancestral nervous systems actually look like. So like imagining what Bilbo's great-great-grandfather looked like based on examining Bilbo, Frodo, and Merry and all of them, it's not that straightforward, even with humans. So we're inferring what the ancestors look like by comparing descendants. And So this is just, I mean, I'm editorializing slightly, but I am not 100% convinced that genetic methods are necessarily the ground truth. So he's based a lot of his work, and my lab actually loves it too, Luis Puey's work, which updated the way the hypothalamus is talked about, which I'll get to, using gene fate maps, which are an excellent way to do developmental studies. So you see, where did the genes that were expressed in the embryo end up in the adult, that kind of thing. And these are excellent, careful studies of this. But the evolutionary leap from the genetic fate map in development to the phylogenetic tree, I think you have to take it with a pinch of salt, but it's hard not to just buy the story, because we all seem to sort of take that. I mean, I'm not a particularly reductionist, but I kind of, even I find myself believing the genetic story when it's presented to me. So I feel like we have to kind of be careful here. And I'll give you an example. I'm not just saying this. Everybody knows about the striatum, and a lot of you will know that one of the classes of interneurons in the striatum is a fast spiking interneuron. We described functionally. But when you stain it, you find in rodents that they are the fast spiking interneurons are paravalbumin expressing. That's a calcium binding protein. There are fast spiking interneurons in the striatum and elsewhere in the primate, but they express calretinin, not paravalbumin. And I was like doing something, and my lab mates showed me this, and I was like, oh boy. Obviously, it's hard not to see the homology of the areas, but somehow the protein has been swapped out. The function is more or less the same. So there's something to keep in mind. And I have a sort of speculative story about how that might work, but that's just a little sort of almost a footnote. Does anybody know what this is? Very controversial. It's Ernst.
I've seen that before, right? But yeah, what is it?
Ernst Haeckel's notorious
Right. Yeah.
phylogeny recapitulating.
Yeah.
Ontogeny recapitulating phylogeny. So that mouthful, for those of you who aren't really in the nitty-gritty bio world, you might not even be aware of it. But Ernst Haeckel in the middle of the 1800s came up with this idea based on looking at embryos that the adult of some ancestral creature is an embryological stage of us. So we kind of pass through our entire evolutionary history while in the womb, basically. It is such a sort of tempting idea if you sort of slap the great chain of being onto the Darwinian story. And even though everyone will say that, well, that's definitely not true, we can show you that with all kinds of data, it's hard not to actually think this way. I find that even in myself. And I find even when people do all this lip service to say, let's not do that, they're kind of doing that on some level. But the way they do it is more subtle, because, so this is on the left, you have the standard story where the entire fish is kind of recapitulated in the early part of the human development. That doesn't happen. But something else which is sort of interesting does happen, which is that the developmental pathway gets more and more complicated in different ways in different lineages. And if you look at this Strider and Northcutt book, so Paul Cisek and Luis Puey's are doing this book club on this recent book on evolution. And one of the early chapters talks about how one thing that someone like Haeckel misses, and a lot of us actually don't think about, is that evolution need not always lead to complexification. It can also lead to simplification. So in some cases, steps in a developmental sort of program, if you want to think about it that way, get removed or simplified or sped up or slow. So the degrees of freedom are quite large. And that's actually something to be worried about even when you do the other kinds of comparative analysis. But they're pretty careful nowadays. So hopefully they're right. So yeah, that's just something that I always worry about. So ontogeny does not recapitulate phylogeny, but it waggles its eyebrows suggestively. So another reason I say that is that when we're going to talk about like the words like medial and dorsal and stuff like that will come up, labeling parts of the brain that are no longer on the medial side of the brain in the adult. The reason they call that is because of this fate map idea kind of run backwards, which is that you kind of see, well, where was it in the embryo at some stage where you can actually make out these different segments. So sometimes when we're talking about these overarching schemes, we're using anatomical language to talk about something from the past, developmentally typically, not evolutionary. But so that you see there's this three different things you have to keep separate, which is the state of the animal now, which you're typically looking at an adult. There's the developmental history, which tells us a whole lot of useful information, and the evolutionary history, which we're kind of, really spitballing a lot of it, but with good principles. Anyway, so as I used the term fate map, it's a super cool concept, and Puez has done some brilliant work on this stuff. So his whole kind of way of thinking is that molecular boundaries are thought to be much stronger than comparatively conserved limits, since they reflect primary causal regionalization features. Hard to argue with that, given how we think that the animal develops. So basically earlier, people would use these sort of qualitative features, often mixing the sort of shape of the adult with the shape of the embryo. And the main example being how to think about the hypothalamus. Why the hypothalamus? We'll soon find out, but if you haven't read the paper. But basically, this left diagram is actually how most people until very recently thought about the hypothalamus. And because of some powerful anatomists in America, it's still quite popular. So I say put old in scare quotes because a lot of people still think this way. So basically, there's this kind of division into the alar and basal plate, which I'll show a better diagram of. And so earlier, when people thought about hypothalamus, they kind of thought of it as like a little sort of outgrowth of the thalamus or something under the thalamus, and that the cortex kind of emerged from the thalamus. So you kind of have this almost a functional perspective on what is more important. So if the thalamus is sort of second in command to the cortex, you think the cortex grew out of the thalamus. But with the help of these genetic markers, now you see a very strange and interesting story. In this mouse brain, it looks particularly strange. So basically, development up until you get to the vertebrates is kind of just like these sort of Lego blocks in a row that sort of added, and then things start to bend a little bit. But you can kind of still see these units. They're called neuromeres. What happens is that instead of just slapping on another thing in the front, somewhere in the middle of the hypothalamus, something sort of goes sideways. It's hard to fully visualize this. I would just recommend looking at lots of diagrams to think about this. So something kind of pops out of the hypothalamus, sort of almost orthogonal, but don't think of that word geometrically, just sort of topologically in some sense. So the cortex is an elaborate specialization and outgrowth of the hypothalamus, which is a new idea. It's a 21st century idea, and it's quite powerful. And it's a big part of what the story that Paul Cisek tells. So basic idea, which I'm really simplifying here, the dorsal ventral division, which you see here in an adult, is arbitrary because of all the turns that happen and does not reflect developmental or evolutionary kinship. And like I said, the hypothalamus is not under the thalamus from an evo-devo topological perspective. It sort of is what produces, it's almost a staging area for the development of the cortex. So what Paul does is, so here I'm just looking at this bit right at the beginning, which is the story of how these very simple creatures, the eumetazoans, EU is Greek for good, so I guess good many cell creatures, were really simple. They had an apex and a base, which was a mouth, I believe. And did these creatures have that sort of mouth anus as the same thing? I don't remember, but so basically they elongated, and there were these two sort of neural clumps: the apical clump of neurons, ANS, apical neural system, and the blastoporal neural system. So you make this sort of elongated tube, and then these group of neurons kind of sink into the skin, the rest of the body, basically. So that's the formation of the neural tube. And then what happens is these two kind of ancient divisions merge, and where that merge is where the future central nervous system is, the head. So this is what I was saying about neuromeres. If you sort of start at the spinal cord, you see these neuromeres in these units. And at the very front, you see this merging of the alar and basal. So these two types of that were originally on the opposite ends of the simpler ancestor merge. And so see, the thing is here, when you look at a diagram like this, it's easy to say nod along, and then you're like, am I looking at a diagram of a currently existing creature or an ancestor? So if I haven't mentioned it, please ask. In this case, they're talking about currently existing creatures in this diagram. You know? But sometimes you'll be talking about a hypothetical reconstruction of the brain of some animal. So yeah, so you're kind of assuming that things that are currently existing are living ancestors or living fossils, even though we keep telling ourselves we're not supposed to do that. But something like that ends up happening anyway. So one narrative that you can kind of pull out of this is that of the different parts of the hypothalamus that explode, the real thing to watch is the pallium. If you Google pallium, you'll see some sort of papal outfit. I think it means cloak. So it was the cloak of some ancestor / developmental stage, but it's not a cloak anymore. If you look at pallial parts of, well, cortex sort of. So Bauplan means like housing plan, I think, in Germany. So there's this basic plan, and how it kind of gets changed in the front of the brain of this tube is the big story here. So I just pulled this from Wikipedia because I find it very hard to keep track of where medial is after a while. So in a shark, so these basal chordates, are pretty simple and precede even the formation of bony fish, is the idea. There you can, the words medial, dorsal, and ventral kind of make sense. So this is a coronal section. And this medial part is here, then you have this dorsal, and you have a lateral part. Eventually they'll talk about a lateral ventral part, and then it becomes hard to imagine why when you look at these flat 2D diagrams. So the story here is that in mammals, you get one sort of kind of differentiation in the pallium, and in birds you get a different one with a different pattern of neuron formation. But in order to like deal with a complicated diagram like this, it's good to have your bearings at least somewhat. So we're talking here about the simple, I think this is like a chordate. So this is, I believe, a kind of reconstruction of an animal that just before the sort of true vertebrates evolved. You can see the neuromere structures, and so these functional sort of labels here, very cleverly sort of placed as if they're anatomical, is the real story here. Although you can kind of get lost in the anatomical details, and then you have to keep revisiting the functional story, which I'll get to. So basically, this kind of like a simpler and a sort of later stage in chordates. I think I'll come back to this diagram again. So yeah, there's other terminology to keep track of. The word encephalon in Greek means head or brain. And so this will help keep, so this is a sagittal section. And so we're looking, side on. And for a while I couldn't quite figure out why medial was up here and where exactly ventrolateral was. But you have to think of these medial parts as both medial and dorsal. That kind of helps. So it's like kind of, if you're looking at it head on, it's sort of, there's this on top and then a swerve around. But yes, so as I was saying, this is a hypothalamic segment. He called, in Puey's terminology, they're the prosomeres, which are sort of like neuromeres. And so you see that rather than kind of tacking on more things like in sequence, like carrying on that neuromeric kind of phenomenon, instead you're kind of going, I don't know what to say, sideways out from this segment here with the pallidum. So pallidum should not be confused with pallium. Pallidum is part of the subpallium. And then you have these various pallial parts, and each of them will go on to do great things. So that's the narrative here.
If you speak Portuguese, it's really easy because pallidum is just pale.
Oh, okay.
That is basically, so it's That's why you don't confuse pallium and pallidum.
Nice. And now, yeah, good to remember.
So Yohan, do you want us to discuss at some point at the end? Is that how you?
You just interrupt me whenever if you want to bring up something in the paper, and yeah.
Yeah, you should jump in. But before that, just a quick question. This is when they say the ancestral and hypothetical vertebrate brain, or what if that's your editorializing of it?
It's me, yeah. But ancestral, yeah.
What does it mean? Is it like these should be the set of structures that have to have been there for your evolution from that original vertebrate brain to when you become a human?
Yeah. Which in practice will look a lot like a fish brain.
Yeah, I think one good way, it doesn't have to be a fish, right? So I think one good way to think about it is essentially you do phylogenetic analysis in which you try to understand from the commonalities of diverse groups of ancestors what provides evidence for some common structures that likely originated in ancestors as opposed to having originated in parallel at later points in phylogeny. Okay. That was the quintessential mode of thinking and a brilliant insight that revolutionized things in the 60s and led to the explosion of understanding. We hope it's understanding because it makes a strong assumption that convergent parallel inventions during evolution are relatively rare. So I don't develop some type of structure. If birds, let's think of birds and mammals. The lines to the birds and to the mammals diverged 310 million years ago, or approximately, that's an estimate, but it's seems current day pretty decent estimate. So if you find something in my brain, in the bird of the brain, the inference is that it was present in our common ancestor. However, another scenario is one in which it wasn't, but it developed through evolutionary processes and evolutionary trajectories of dynamics or what have you. And it just so happens that you now find it in current day specimens, which is all that you have access to, like Yohan was saying, you find those two things. And so insofar as the logic of the unlikelihood of having convergent evolution stands, this is a solid argument. I don't know if this makes sense without a chalkboard, a blackboard, but essentially it's if we had a board in here in front of us, everyone would clearly see it. But if you just maybe can sketch it on a piece of paper in front of you. But one of the things that is happening in the last 5-10 years that is going to throw everything in disarray, potentially, is at least the possibility that there is more room for convergent processes, meaning that de novo type of things, like you have a certain eye and some other very different creature has that same eye, but there's no common ancestor. How could that be? Well, you two invented the same eye. And how unlikely that is, or how likely, is really difficult to ascertain. And we are really bad at our intuitions of likelihood and unlikelihood in biology.
So Paul
because we understand so little. I'm sorry?
Paul has an example from this paper.
Okay, I see. Because I didn't have time to read it. But yeah, there are plenty of examples that people give. Go ahead.
So apparently in birds, there's a classic paper. Well, it's not that old, but you have to have, like, all vertebrates have a basal ganglia, but not all vertebrates have a basal ganglia loop, apparently. So the output from the output structures of the basal ganglia were typically to tectal and sort of control output. So it was almost like an open loop structure. But this connection going from globus pallidus to thalamus and back to cortex apparently evolved separately in birds and humans, which is very interesting. And you can tell why that inference was made if you look at this diagram here, right? So if you don't see it in lizards and amphibians, and you do see it in birds and mammals, then you're like, well, the common ancestor must not have had it, or it was lost in the earth. So how to tell whether
there's also the losing, right?
Yeah.
Like I had something, but I lost it. Yeah.
Yeah, I don't know how you can tell those two apart, but there's all these other kind of like people aren't only using the brain to make these trees, nor are they only using genetics. So one hopes that it's almost
But
like
Yohan, there's a lot of work on cephalopods that are very diverse group of invertebrates, right? You know, like octopus, cuttlefish, squids. I'm blanking, but essentially, and they are very diverse, including some that are thought to be ancient and some that are thought to be really modern. And if you look across a very large group of them, the ancient ones have things that the new ones don't. And I mean, I can't come up with it in the top of my head because I don't remember all the details, but essentially there's pretty convincing evidence that they lost many features. The newer ones lost many features for whatever reason, right? It's always risky in evolution to like, why did it lose that feature? But nonetheless, I think it's very well documented. It's fairly well established.
I think, you can think of, if you think of genes as traits, which you kind of like bit segments of DNA as traits, like onions have more genes than humans. So in some cases, the sort of path of complexity pursued in one sense was at the level of protein regulation or something like that. In other places, like huge chunks of the genome have been lost. In fact, where did I see this? Luis, you might remember seeing this paper. There was a paper saying we have to be careful about making the rodents our only lab model, because in some ways rodents are quite unique in the things that they have lost compared to other vertebrates.
They're so specialized, right? And in a sense that they don't become great representatives anymore. Going back to Kale's question here, it's exactly the nautilus, the nautilus, or whatever, how you pronounce it properly, but at least in English, the nautilus is again, I'm far from an expert in this. I'm trying to remember not to get it wrong, but the nautilus is a very ancient type of
somewhere here, right?
It's quite ancient compared to other much more modern octopuses and other kinds of cuttlefish and squids. And so that's actually one example that they look at several groups and in groups and out groups and their relationships and behaviorally and what kind of habitat they inhabit. Is it all throughout the water column? Is it they move along the day or whatnot every day? They do this or that. So they have a whole lot of behavioral repertoire and linking it to a lot of properties of the nervous system, sensory motor properties and all sorts of things, which is really fascinating. So this possibility of losing features and of having de novo or quasi or what have you, de novo types of situations complicates the story tremendously. And so it really, if it's really true, what some people seem to be finding now, that there's more room for evolving again, certain kinds of paths reoccurring, then that whole logic of that we build all these things sort of falls apart to a good extent. So we would be in big trouble.
It's a very interesting possibility. So this is the kind of central functional story that Paul wants to tell, which is nice and simple. Although when you first look at this, you'd think that the ovals mean the organism, but there's like food on the inside of one of the ovals. So they're abstract functions. And more or less, you could think of it as like, so there's regulation of whether you should eat whether there's food in front of you, do you eat? And, you know, as you eat, your nutrient levels go up, and then you feel satiety, and 5-HT goes up, and then that can inhibit hunger. So there's that loop. He talked about this with us when he came, and but then you may have some reason to leave where you are. It could be avoiding a predator, or that there's nothing useful nearby, and then you need to make these larger movements. So there's this, I had this song cropped up in my head when I was reading this. It's the should I stay or should I go? It's the simplest functional dichotomy. And the story that Paul tells in this paper is about how the stay can become various things, and how the go can become various things, and different in different lineages. So that would be the functional overlay on top of the structural story. So in the early chordates, these are pre-vertebrate, but they had a notochord. You see this kind of 3 steps, which I believe even I get confused sometimes. I believe these are existing creatures, where initially you had just this frontal eye patch, which was basically a light sensor. There was no lens, you couldn't really form an image. And you had ipsilateral projection. And last time he was here, we all found it very hard to believe what he said, which I'm about to say, which was that the crossings from one side to the other did help you escape. So at the time, I had no idea what he meant by it, but then I thought about it a little bit, and I realized that because muscles can only contract, then if you imagine something that needs to get out of the way and not go towards something, you kind of want it to contract in a particular direction. So that, I think, is the logic behind this crossing story, so that this crossing in step B improves the ability to escape. Now, I asked Paul actually last time, but what about creatures who didn't have this crossing? Could they not escape? They could, which becomes a huge computational question then. How did they do it? Because if you look, you know, there's all these crossings and then crossings again in the spinal cord, or before you reach the spinal cord. So then it becomes confusing. once you have 2 of those crossings, my logic kind of fails at that point. And then you need a model, you know, because ultimately simply contracting all your muscles at the same time is not going to do much for you. You're not going to move really. So you need to kind of do a bit of that too. So anyway, so the idea which he kind of uses this as the toy kind of place to put it in is that
Yohan, confused. So Paul's I remember, I just about remember.
None of us got it.
I just about remember what he said in that lecture. So he says decussation is necessary for escape.
No. He didn't say that. No, he said that it improves escape.
Improves escape.
So the problem is that the story for the decussation is almost too good, right? Because then you're like, well, how does something without the decussation escape at all? So he said, yeah.
Improves it with what? For what? Like,
I don't know.
Is it energy, like metabolism, energy, like in terms of what?
You know, it's not enough detail really. I've asked him about it twice actually. I still have not like got a clear sense, and I think because we're modelers, we're immediately thinking, what am I gaining or losing from doing this kind of thing? And from a biophysical or biomechanical perspective, I don't completely see it, especially if I get this other crossing.
Yeah, when I read it in the paper, I thought that's just too convenient of an explanation.
Yeah,
it's got to be more complicated.
I'm sure it throws off a lot of readers. So I even for this draft, I told him, I don't get it, but one sort of functional story which doesn't necessarily apply to this mechanism, but is an interesting thing, is that when you have predators, then sort of summing their sort of intensity, like let's say all you're doing is using light intensity of something large to say, I need to get out of here. You can average the light intensity of something large to escape, because you're going wherever the average is low or something like that. but that is not particularly useful if you want to pursue something, because the average of 2 pieces of food is nothing. So he kind of uses this to motivate the idea of the winner-take-all kind of behavior starts to become quite useful when something like predation happens. So to become a predator, you'll need to actually not just do this summation or averaging or lumping. You kind of have to pick one or the other. Otherwise, you'll only get the worst of both worlds. Right. So what this circuit is kind
Can
of.
I jump in real quick, Yohan?
Oh yeah, go for it. Yeah.
Sorry. Just while we're talking about the crossing, uncrossing thing, I often wonder, I haven't actually asked Paul this directly, if it relates to an idea that Valentino Breitenberg espoused in his book Vehicles. And if people haven't read it, it's a real fun little book. It's this, it's only about 100 pages or so, but Breitenberg was one of these incredible larger than life characters, right, that could think really deeply about implications in really, really simple organisms. And the argument he was trying to make, it was really, I think to read it in the tone that it was intended, you've got to be thinking about, what he was trying to fight back against, which was this notion in a lot of ways of like what was around at the time of behaviorism, that, simple mechanisms weren't particularly exciting or interesting. He was trying to say, look, you can have really, really simple mechanisms in little vehicles where we know the kinds of connections between the sensors and the outputs. They can give rise to things that look extremely interesting on the outside. They can be, really fascinating, like, liking and wanting was one of them. But then you could also have, if you have a little threshold in there, you can have deliberating and decision making, and there are all these really fun little examples. But in his kind of framework, the first vehicle he makes has a sensor that leads directly to a motor effector, which in this case is the driver for a wheel on exactly the same size of its body. And so then if that little vehicle sees a thing that it likes, in this case, it was light, it wants to move towards light. If it's away from light and light is on one of the sides where it wants to, where its sensor activates, it'll then turn the motor on that side and it'll sort of rapidly crash into the light, like someone sort of like steering, into a ditch. And again for the other side. And so it looks like it has approach-like behavior. And then he argues that if you wanted to avoid behavior, what you need is crossing fibers that turned on the motor for the other side, so that if you saw something you didn't like, I can't remember what the example was in his case. Let's say that now it's, chocolate. If you see chocolate on the right-hand side, now it'll turn on the motor for the left and you start moving away from it. And so I think in part, if you start with this really simple premise of, the first organism in a long sequence of organisms having sensors that attach to motors, it's really easy and simple to come up with a sensor on the same side equals approach and sensor on the opposite side equals avoid kind of an architecture. Right. Now, obviously, that's not the only way to solve that problem, and it would depend in a lot of ways on how the system was organized in the first place and whether or not the movements were, in fact, something like a motor or more like a contractile peristaltic wave, which you're probably more likely to see in worms and things. And so it's very overly complicated. So I think I'm on your side there.
Yeah,
it needs not be the case. Yeah,
I'm not saying decussation doesn't have that might not be useful. It's just that I feel like the claim, the causal claim for the functionality, it might not be like true, right? Like it's like
need to be as selective as that.
Yeah. So that's what I'm not sure about.
So here's I understand the logic for the car or the wheel thing, right? But mapping that onto how locomotion works in the ocean, there's something about the biophysics of that. The mechanically, I don't see how, because of the fact that excitation of a muscle fiber will only lead to contraction, and contraction is not like the rolling of a wheel. In that, once it's done, it's done. So you need the opposite contraction to do any of that. So
I think, so I think that so go ahead, Keel.
Yeah, so I was just thinking, imagine if you have a little tail that's acting like a propeller at the end of the tip, right? And then you just have the shape of the animal. If the animal is a C shape in one direction, it's going to go that way. And if the animal is a shape in this direction, it's going to go that way. And then it functions similarly. It's just exactly the same if you just contract one side of the body.
Yeah, because I was gonna say is that the one thing that I don't know enough by any means, but if you're really interested in this question, there's a lot of work in fish about this C reflex, literally like the C that Kay was doing, like the cupping, and the fact that, I don't know if anyone have you, I'm sure Mac has, and anyone who has grown up and lived by the sea and whatnot, catching fish small, like as a kid, it's impossible. I mean, it's like so difficult, right? They're so fast. I mean, within 5 milliseconds, that thing just moves. And so it's practically impossible to catch it. And so the argument is not that it's only feasible that way, but one that it conferred an advantage in terms of the speed of this reflex. And this guy that works in, I think, UCL, his last name is Branco, is Tiago Branco. Branco is Branco. He has beautiful work on approach and escape across many vertebrates, and he has a review in ticks, in Trends in Cognitive Sciences, that discusses these circuits and in a lot of pointing to a lot of work that talks about it. So anyway, so I think that anyone interested, it would be fun to see how does exactly this circuit work, and I'm sure it's been mapped out, because it has to be a fairly simple circuit to function within 5 milliseconds. I mean, it can't be, but oddly enough, it's a circuit that can be modulated by all sorts of things too. So it's not just completely automatic, not even that one.
Yeah,
so it's
yeah,
very
yeah.
cool. Yeah, it's a fascinating, simple thing that is actually turns out to be quite sophisticated.
Yeah. So yeah, I just took some, like, I was just trying to make some sense out of all the details. But the story here is that in these early hypothetical chordates, you see the merger of those two groups. You see the posterior neural tube kind of becoming hindbrain and spinal cord. We didn't really talk about this, but this is like a fairly simple idea that the dopamine level, so just the tonic level can convey whether to eat or whether to explore. So at this point, there's presumably, or the idea is that the tonic, the phasic response hasn't evolved yet. And you have this dorsal allostructure, which is the prototectum. So the tectum was like this early controller, like some, ancient aristocrat or something that used to be doing a whole lot of work. And one of the stories of this whole development is the sort of gradual loss of control by the tectum as a result of all this other stuff, which starts as a modulator or helper. It's like being like a coup by the vizier, kind of, So in these diagrams,
Can I ask a question about this? In these diagrams, what's going on with these explore, exploit, escape arrows?
So they're basically, so the other diagram was like a top-down kind of view. This is a side-on view, but it's the same crossing that he's talking about. But instead of going just to muscles, they're going to these structures that are pattern generators. So some of them.
So these are labeling the circuits?
The circuit, yeah.
Yeah, I mean, this is part of his phylogenetic refinement. So they are becoming increasingly, for lack of a better word, complex.
So the pathways do exist. Whether they only do what is labeled on there is the big debate, right?
So I just wanted to follow up on Ryan's question. If you look on figure A, you see how from the same region it has explore and then exploit are the same connections, right? From that region to that region. So there must be a different kind of activation or different kind of signal from region A to region B. That's an exploit signal and an explore signal. There must be something that we're just glossing over. That's what the explore versus exploit label are referring to.
You see that that gets enlarged in the second, in the space B, right? In the picture B, you see that explore versus exploit. You see the difference in the connection. So it starts off with something that looks very similar, exploit and explore being from the same space, basically that conceptual space that Yohan had earlier. And then as the structure evolves, you see in the bee with that maroon or whatever, purple versus blue changes. You see that it actually might start at the same starting point, but then it'll have to travel through a few different nuclei or something like that before it reaches the reticulospinal nuclei for action.
Yeah, so in B, it's clear that if there's more like neural firing at the explore tracks neurons, then the animal is going to be exploring. It's just which is more active, but it's still not totally clear to
me. Think of it as just a proto version, in the sense that it's already starting to do something like exploit or explore, whichever is the later one. It's starting to do things more than just exploit. It's doing a little more exploration of that space, even with that proto circuit, and then it gets greatly expanded, right? So
the other possibility, which is also speculative, is that, so I guess, Kale, what you're asking is this structure, can you see my mouse? Is this structure differentiated or not? Like functionally differentiated. He's not drawn it as if it's differentiated, and then you would ask, well, if a bunch of things are converging on one neuron, how can that one neuron do 3 different things? It can't, unless you say that the temporal pattern of input can produce very different bodily movements. I mean,
in part
I would
A,
not assign this as a project to anyone, but it's doable.
In part A is supposed to be some
here.
Right, but it's supposed to represent something that is prior to chordates, or is that what it is?
This is even earlier than this, basically.
Right. So it's like, basically he's trying to see, like, essentially, when does this, it all has to start somewhere, or basically if you track back down the history,
right.
It's his kind of suggestion, right? His broad suggestion of some possible earlier version of it, but presumably highly speculative, right?
Yeah.
Including the newer version, the part B and so on. It's all highly speculative. I think that's an important thing to note here.
Some of these patterns we do see in laboratory preparation. So it's saying that it only does one particular function.
Right, but Yohan, as you said, like, the problem is, I mean, I think in the brain, the problem is always like when you put a label onto something, that's when it goes wrong. I think that's, in my view, so when you put a label, it's when that you hugely simplify, but we can't do anything without putting labels.
Yeah, you just assume that they're all tentative.
We have to maintain some flexibility, mental flexibility, understand that it's a rough guideline.
However detailed this seems, you have to treat it as like only the first or second or third term in some infinite series. So this little bit here is what he focuses on in the new paper, and because of this like straight line here in the diagram, for a while I was like, what am I looking at? Like, because it was a little hard to find my bearings, and then I dug up the older pictures. But so by this time he's got quite a functional localization. It does involve broad circuits, but it's still fairly like localized. The idea being that this the medial pallium, medial and dorsal still in these simpler vertebrates, which is the future hippocampus. It's part of what reading this when you're not familiar with all these anatomical terms is like, it's like watching the prequel to a movie. It's like, oh, this person will grow up to be Darth Vader, like that's the feeling that you get when you read this. So, watch this bit. And then this dorsal pallium, watch this bit very carefully. This tiny newcomer, which wasn't even allocated a space, will do a whole lot. The other interesting thing in this story is that the amygdala is almost like not a part of the story, in the sense, so it's sort of like when you have all these striations and bends and whatever, there's always going to be some sort of boundary condition, which is like when the things that are different meet, there's like, it's almost like a little knot that needs to be tied, and the amygdala is that knot. The other knot sort of is out here, and it's adjacent to the olfactory cortex, or the bulb actually. And in fact, He cites Senneides, but he doesn't like Senneides' evolutionary story. But there's this idea that the cortex grew out of the two ends of the hippocampus, or this medial pallium, which is the para-olfactory trend and the para-amygdala trend. So yeah. So these are, we're still like, so we've reached the vertebrates now. The amniotes are the mammals, birds, and the anamniotes are, I forget what, fish and amphibians.
Fish and amphibians, yeah.
Yeah.
So what's sorry for all the basic questions. What's being
please.
used to differentiate these areas? Are these functional differentiations
So
or molecular?
this is an
or
excellent
what?
question. It's not basic at all, because and this is again why it's such a difficult thing to summarize, is that in part the functional story comes from rats and monkeys, like just lab animals now, right? And in part you're inferring, and in part you're looking at the genetic fate maps from development. So all these things together are forming this image. Sometimes like following the logic back is tricky.
I think one of the main things, though, is they take present anatomists take present animals and dissect the brain, and then physical boundaries are visible on staining, or just through gross dissection without any staining. You'll see an edge to the amygdala where there's like, it's not membranes, but there's collections of cell bodies with then no cell bodies adjacent, and you can actually see them in gross dissection. Like if you take a sheep brain, you can actually dissect out the amygdala just through looking. And a lot of these lines are just from present day animals where those edges to the structures are.
Yeah, like it's really
yeah.
easy
So I was going to say,
yeah,
I was going to say the answer is essentially structural, right? So we see via a series of techniques, some genetics, some all sorts of different techniques to highlight structural differences of some sort or another. Sometimes they paint slightly different pictures, and that gets also complicated.
Which is why, like, this is the place, like this, what Paul is attempting, what everyone is doing here in combining the genetics and the evolution and the function, is where like from a physicist, my background in physics, right? So when I first heard about disputes in nomenclature, I was like, what? How could that possibly matter? But as it turns out, it can matter quite a bit if you're asking about homologs. Like right before I joined grad school, there was a consortium of bird neuroanatomists who renamed everything to show part of what is the point of this paper also, which is that certain parts that people thought were descendant of one part were not. And I mean, this has a lot of impact on what you think is the homologous or homologous part of two different species. So
are you talking about the Fernando Nottebohm? Is that the one?
It's 2005. I was looking at it.
Yeah, the bird story is fascinating because it was a completely antiquated way of thinking about it. It got completely changed. It was completely redone, essentially from the early 2000s. It revolutionized how we see bird brains.
Yeah,
and as there's been an explosion of research, there was literally lots of things that were just wrong according to current view that got rewritten. No, this is actually something part of a different part of territory. It's a different class. You're not in Asia. You actually are in the Americas here. So, oh, okay, that changes things a little. So
what's happening now in this decade or previous decade is this revisiting of what the hypothalamus is, Puey's work and stuff like that. So it's a similar kind of thing is happening in that too. So yes, so now we're starting to see these functional stories about VL ventrolateral pallium. I remember why I pulled this out, because of the tectum is still in charge in that red bit that I underlined there. Yeah.
I just want to point out that this, is also true much more recently evolutionarily. I mean, my current PI, Gaurav Patel, It suggests that even rhesus macaque monkeys don't have a TPJ, a temporal parietal junction circuit that humans have. So these splits get, a lot are even much later evolutionarily.
Makes sense. Yeah.
A and B, the hypothalamus. So, for example, my first scientific mentor, Ray Silver's lab, just published a paper this year that the hypothalamus has portal systems that have been sitting there undiscovered since the 1930s. So the first portal system was discovered in 1935, and the next one came out this year.
What's portal system?
So portal system is direct capillary connections between a nucleus and an expressive region. So it allows very small nuclei to create high concentrations of neurotransmitters that would be out of the league of, normally out of the league if they were diluted into the main bloodstream, rather than having capillary connections.
So it's like a way of creating like microenvironments.
Yeah, exactly.
Right, I should look into that. So yeah, I pulled out this excerpt because I'm pointing out that even though we're starting to see these interlopers from the hypothalamus starting to go and tell the tectum what to do, the tectum is still in charge. So then you see this. So again, like he's piecing together all these different pieces to say, so the VL, PL, and subpallium recognize the impetus of hunger plus edible. And the reason he's framing it this way, slight anthropomorphization, is because he wants to say that the concept of affordances will start to become much more visible anatomically, basically. So yeah, so this now we get to lizards. So here's where he starts to kind of really stress this notion of affordances, and here's where I also kind of lose the plot a little bit. So this new bit kind of interposed between these older pallial structures is the dorsal pallium. And so what's interesting here is that new visual signals are starting to be a big driver. And by this time, the sort of shift from the sea to the land has taken place, and there's new selective pressures. So here's this proposal that the DPAL is used to detect affordances. And he mentions that key stimuli are very old. The key stimuli is the idea that there's just this sort of almost like a lookup table between some stimulus and a response. So it seems like it's sort of fuzzy to distinguish between something like that and an affordance. It's a matter of sort of degree, I suppose. I was reading about key stimuli elsewhere, and I found some online encyclopedia that said that when the concept of a key stimuli is presented, it seems like a highly specific thing where this particular stimulus will produce a particular action, but such a thing has never been found. In however simple the animal is, the key stimulus is always a category of stimulus, which from our sort of computational perspective is no surprise at all, because there is no such thing as a distinctive stimulus. Everything is a member of a category.
I mean, you can wonder why the C. elegans people, after having mapped everything, are still unable to figure out, right? So,
yeah. Right. So they're talking about how these MPAL regions are kind of helping the tectal guidance system, and this is partly because of like this loop that I talk about is coming in the birds and the mammals is not yet formed. But his idea is that one type of affordance, this is the classic sort of Gibsonian frame, is that the animal doesn't need to represent the world. Instead, it can just use the optic flow to decide whether there's an overhang or not. I've never seen a demo of a model that uses optic flow so easily. They make it sound like it's super simple. But I always wonder, what about changes in lighting? What about like, how do you latch on to what it is that is supposed to be flowing? So that's a computational question. But when it's presented this way, you're like, yes, very easy. But maybe it is easy. I don't know. So then we get to the point where, so basically these amphibians have emerged from the sea and they're starting to live on the land. And initially they're living diurnal lives. I believe the fish also did, the ancestors. And at some point, the mammals start to become nocturnal. And this is partly because the predators are diurnal. And so a lot of changes take place along with this. And basal ganglia loops seem to happen around this time. So this DPAL, this tiny little thing, this newcomer, becomes the whole neocortex. And has this unusual organization. So there's a completely new way of populating neurons inside out, where you start from the subventricular zone and you kind of, so layer 6 is done first, then 5, then and so on. 1 is always there. And the way that the thalamus actually enters is different. So in reptiles, apparently, the thalamic input comes tangentially, like from the sides. This radial projection is new for mammals. And yes, so here's where this novel thing, and here he doesn't point it out that it's a separate development. Either it's separate or the ancestors lost it, in which case the story doesn't work. But it developed so that there was this additional output of the pallidum, which is globus pallidus, ventral pallidum, the output stage of the basal ganglia. Instead of projecting downstream and out in a kind of feed-forward control, projected to a ventrolateral subdivision of the thalamus, which itself projects back to the neocortex. This should ring some bells. It's the classic basal ganglia thalamus cortex loop. So now we have this sort of reverberation, which he'll suggest is helping with selection, action selection, things like that. So there's a whole lot of detail here that I didn't actually try to even summarize, because you should just read it yourself. But the end of the story is that now that one little sliver, it was this little sliver over here, now this pink bit is now, it balloons out around and swallows up everything else. You kind of have to look at a lot of diagrams and imagine it for yourself. In fact, if you're curious, brainmaps.org allows you to look through serial sections of human, monkey, rat, ferret, I think maybe. So you can actually go, I would recommend going with the coronal section, but you can look sagittally also. And then you can, and it's labeled, so you can see where the different areas are. And you'll see there's a Nissl stain and probably a CH stain too. So you can see how easily these areas can be distinguished. So yes, everything explodes. And here's where areas that were kind of just modulators of the tectum using fairly simple combinations of stimuli are now doing this sort of basically, I don't know whether he calls it that, but kind of conjunctive coding. And his idea is that this takes two forms. Here, I don't actually buy the story, or at least I don't think he's provided enough, evidence for it. But he cites Barbara Finlay's work about two sheets of the cortex, like sort of two styles of cortical processing in this. One is more spatial and topographic, and one less so. So the sort of the what-where stream, basically. But I don't think that that's the right way to describe the difference between these dorsal and ventral streams of the cortex. But it's an idea to think about, I guess. So what monkeys seem to really develop is this ability to handle. So what initially was, like, if you think back all the way to the pre-fish kind of thing, there's not a whole lot of dexterity that needs to happen. But now you get all this independent control. So the types of movements that you're capable of are quite different. So handling itself becomes a whole lot of different things. So this is part of the stay mode, right? Which is, now that I'm here, I can dig, I can move things around, I can bend branches down. There's lots and lots of things one can do. And the other thing is that mammals use their eyes a lot more. So this is, I think they're starting to and also other sensory signals, because so earlier, now we're talking about primates, right? So by this time they're back to being diurnal. So an interesting kind of theme here, which I would like to know more about, is this sort of nocturnal interregnum, which caused kind of a loss of certain connections, which then were kind of reconstructed in a different way. That's how I see it. That's my fuzzy way of thinking about it. But so they're back to being diurnal with this new set of connections with these new areas. And so their earlier mammals were running around and sniffing. whereas anthropoid primates are looking around more. And what this reminds me of, I'm so sure some of you have seen this, is like they found something like place cells and maybe even good cells for monkeys sitting stationary and looking at things. So initially you'd be like, well, why should the visual system produce anything like a place cell, which in the rodent is, they have to be moving around. And here's a way to think of it. Is like, I even have said this to some of you, that when you're comparing the rodent data to a primate or a human, it's almost like human attention is the mouse, is the whole mouse, like like running around. And he's kind of on the same page. And if I say that to him, he'll totally like know exactly.
And human retina is the entire visual system of the mouse too. So
Helen will like that, yeah. But yeah, so basically you go from having to actually be near the food in order to realize it's food, to like saying, oh, there's a nice juicy fruit over there, I shall make my way there. So one of the themes he kind of brings up is that over the course of this elaboration, you develop the ability to latch on to longer timescales, which I think is very important. Not clear to me that you can map it onto anatomy so easily, but it's a good thing to think about, which is that one way in which animals change is the timescale at which they're able to retain information. The other thing he does bring up is the idea of like second order affordances, which is prediction. It's like, if I do this, such and such will happen. And then think about that. And we know that anyone who's interacted with monkeys knows how smart they are. In India, we've had a lot of encounters with them. But, so there's two kinds of expansion. So you could say that possibility space expands with the primates a lot, both in terms of what the consequences of one actions are and the amount of history that you're integrating and the amount of future time that you're integrating. So that's kind of where the story ends. I don't think I've, oh yeah. So basically you go from this sort of funny little tube where the ancestor of the cortex was not even present yet, really, to all of this stuff. And so, yeah, I like this story because I think it's sort of an interesting thing to investigate the consequences of, even in primates, which is that there was this reduction of tectal control, but not elimination. So when we, all these cortical chauvinists half the time, right? So are we missing something when we ignore some of these older structures for control? That's something that one can actually look into.
You can easily look into that, right? Like, yeah. All I have to do is just like take my own favorite examples, right? If I cut off the superior colliculus or your pulvinar or thalamus, you might see something. It means nothing. The animal's behavior is going to be completely done.
So that's a good point, actually. So in case I didn't say it earlier, superior colliculus, inferior colliculus were the tectum. So very much still present and really crucial. Like you get complete sort of perception action loops from simulating in colliculus, right?
Yep. In fact, I think Mac recently shared a paper from Ziad Hafid's lab, actually showing how you can even have like very coarse-grained object processing in superior colliculus, like in the sense that it doesn't have the fine detail that you will have in say the cortex, but it does it much earlier. So it's sort of like it's providing a gist for the entire system to work with, and then you can refine it later after a few hundred milliseconds with a much larger, more refined visual cortical circuit. It's doing all those bizarre things, even visually, not just visual neurons in superior colliculus, even visual motor evoked neurons have that. So it's very remarkable. It's like across the entire nucleus of the superior colliculus, you observe this kind of object processing.
So I mean, that's true of other, fast circuits as well. I mean, so like Moshe Bar's, proposal of a fast visual circuit into OFC to prime temporal cortex for
the whole point
mental
is
attention.
There could be fast gist circuits everywhere, including like, the TRENS model, which makes use of those things. But that's not the idea here. The idea is, if this doesn't exist, all that fast processing done by the cortex might actually point to be of nothing, because you're really not going to behave. That's the whole point with the tectum having a much higher priority than, say, like what the cortex does. Yeah, I'm trying to be a subcortical chauvinist here, but it's okay. I think it's much deserved and required.
I mean, the other not to, okay, I'm not going to say not to beat the drum, since I'm going to beat this drum all the time when we're discussing this type of thing. But that there are other areas, especially in the hypothalamus, like the suprachiasmatic nucleus, that are entirely intact between the difference between species and the suprachiasmatic nucleus is zilch. I mean, it's a completely preserved thing. And you can understand why, because if you screw up even a little bit when you start living long enough that seasonality matters, you are utterly screwed. Or if you miss a dawn or a dusk, whether you're nocturnal or diurnal, you're totally screwed.
Yeah, that's
Yeah, but I think this is an interesting point, but also remember that if you take the amygdala, which is a kind of also a quintessential example of a highly conserved structure, it is quite different across vertebrates. And in a sense, I mean, it does seem to have a central amygdala and other parts of lateral amygdala and whatnot, but it's not something the way you described in your case, like that nothing really changes, like zilch of difference is a different story there. It's sufficiently challenging that keeps researchers trying to map those differences and populations, different types of populations of neurons and all sorts of chemical genetic types of markers that differ and concentrations and whatnot. So it's really interesting that the converse is also true.
Yeah. In fact, that was one of the things in the first project I did in Helen Barbas's lab, was involve the intercalated masses, which are sitting between the basolateral and the central. And there's a story that you tell when you read, Pare's work and all of that rodent stuff, that it's very compelling, right? It's a nice little circuit, but doesn't work in primates. It doesn't work like that. And because those are inhibitory neurons, this fits into a kind of theme that I've noticed, and I haven't revisited in a while, but I'm pretty sure there's a lot of stuff and if you guys know of it, inhibitory neurons seem to change like faster and more like unpredictably as you go across, like when you go from species to species, like where some excitatory population might seem pretty homologous, the pattern of inhibitory innervation changes faster. That's the impression I get. And I think it's a huge place to explore.
I mean, that's one of the interesting things about the SCN. And the SCN, by the way, is one of the very few parts of the brain where it's been experimentally demonstrated that you can pull it out of one animal and put it in another animal, and the other animal will phase change to the donor animal.
Is that true? That's amazing. That experiments have been done?
Yes, Ray Silver. That's also Ray Silver. Ray Silver in the 1970s.
That's insane.
That's wild.
And so not only that, but there's also a distinction. So you can also put the SCN, donor SCN, in a bag rather than having it electrically connect to the rest of the brain. And there is actually a set of behavior, that circadian behavior, that will be recovered by the SCN in a bag, and a different set of circadian behavior that will only be recovered if you allow electrical connections to form.
The bag sits where?
In the third ventricle.
Like,
I see. Okay, I see. So,
connect. Okay, so that relates to that capillary thing you were saying. So some of the effects of through the blood, is that it?
Yes.
Okay. Anyway, yeah. If any of you know of any papers focused on inhibitory neuron development or evolution.
So that was the other side of it, which is the SCN is an entirely, over 90% of the cells in the SCN are inhibitory.
Okay,
so
it's
the
a
SCN
well-conserved inhibitory system. Okay,
it's an entirely inhibitory system that's extremely well conserved.
Right. I guess the cerebellum has that too. You didn't mention it. Paul mentions it a bit, but yeah. So that's all I had to his take-home message you can look at. So that's all I had. Does anyone want to jump back to any of the figures or expand on anything?
I just wanted to say one thing, which is, is we think of science as very contentious and there are a lot of disputes. This field of comparative anatomy, neuroanatomy, or how the brain has changed is an order of magnitude more contentious. Sometimes they literally scream at each other, not in person, but in their papers. And they literally want to, they seem in the paper to want to kill the other. The things that, I didn't want to say this on camera here recorded, but I didn't mean that. What I meant was that metaphorically speaking, they get very worked up. So because they have very, it's a little bit one of those things, right? We all know that all that we do is sort of under